Hello, everyone. First of all, thank you so much for being here today. My name is Adhvaith Sridhar, and I'll be discussing the research I've done over the past few months in using genetic engineering to enhance the immune pathways of certain plants. So when we talk about genetic engineering, oftentimes people think of it as something exclusive to scientists and researchers. However, this isn't always the case. Genetic engineering is very relevant and can be used to solve day-to-day problems. For example, the recent population increase around the world, as shown in this graph here, has caused an increase in food demand as well. However, agricultural scientists and farmers alike are having difficulties increasing their crop production efficiency because of a plethora of environmental factors which inhibit crop growth. For example, frost, drought, and pests and pathogens all decrease crop growth efficiency and can cause a decrease in food production, which is opposite to what we want to see. We want to see an increase in food production in order to meet the increased food demand. So the problem that my research aims to address is that pathogens cause crop loss. And pathogens cause crop loss in a very severe manner. So this isn't a small problem either. In corn, about 40% of the crops grown each year are lost due to pests and pathogens. And in wheat, about 30% of the crops grown are lost due to pests and pathogens. And this amounts to about $220 billion. So it has a very significant economic impact as well. You might be wondering what genetic engineering can be used for to combat this problem. And the answer is that genetic engineering can be used to enhance already existing immune pathways in plants and make them more resistant to bacterial infections and disease, such as pathogens that decrease corn and wheat crop growth. So the specific immune pathway that's of interest to my research project was systemic acquired resistance, also known as SAR. SAR is unique in its ability to confer localized and globalized resistance to an infection. What this means is that when a bacteria infects a plant at a certain site, most immune pathways will only be activated around that area. However, in systemic acquired resistance, immune responses are activated throughout the plant, and this preemptive measure makes it of interest for researchers and agricultural scientists to study. Systemic acquired resistance is induced by a molecule called NHP, and previous research has shown that increasing the amount of NHP in a plant can actually increase the effectiveness of systemic acquired resistance. This ability is the focus of the first part of my research project, in which we examine the pathway for NHP production. So we've found through previous research that NHP is produced in 3 steps, from being converted from L-lysine to dehydropipiculic acid to pipiculic acid, and then its final form, or N-hydroxypipiculic acid. And these steps are catalyzed by a series of enzymes, which are listed below the arrows here. And these enzymes are produced by genes that are named above the arrows. So in order to increase the amount of NHP production in a plant, we need to increase the concentration of these enzymes in the plant itself. And to do this, we must overexpress the genes shown above the arrows. However, an important piece of information is that only the first and third enzymes must be increased in concentration in order for NHP to be overproduced. Previous research done by Dr. Elizabeth Satley at the Satley lab at Stanford University showed that the second enzyme, called reductase, is not required to be overproduced in order for NHP to be produced in increased amounts. So the gene SARD4 is not of interest to my research project. Only ALD1 and FMO1, the 2 genes that code for their respective proteins in increasing NHP levels in a plant, will be of focus in this project. So previous research done in this field has tried to enhance systemic acquired resistance by increasing the levels of NHP in a plant in two main ways. First, they've kind of bypassed the entire genetic aspect of it by just injecting NHP into a plant and supplementing it with that molecule in order to make sure that NHP is truly the molecule that enhances and induces systemic acquired resistance. Second, uncontrolled activation through transient expression of systemic acquired resistance has occurred. What this means is that researchers take edited genes, which have the ALD1 and FMO1 gene sequences on there. And as you might remember, these sequences produce the enzymes that catalyze conversions of other molecules into NHP. And these genes are injected into a plant and expressed once they've been injected into a plant, almost instantly. In controlled activation, however, researchers would put in the edited gene into the plant. And instead of the edited gene being expressed instantaneously, it waits until an external stimulus activated by the researchers actually starts the expression of that gene. And this has not been done in this particular scenario, which leaves a gap in the field that my project will aim to address. So more specifically, the question that prompted my research project was, can we control the activation of an enhanced SAR response in Arabidopsis thaliana? And Arabidopsis thaliana is the plant that we use in my project. So with a little bit more detail, here's how controlled activation works. So the edited genes would be inserted into the plant. They would remain dormant or not expressed until they were exposed to an external stimulus, and in this case, a heat shock. And then the edited genes would be activated. In this case, NHP would be produced and systemic acquired resistance would be induced in an enhanced version. So the first step that we did in my research project was to create the genes and the edited genes. And we created 3 different constructs, 2 of which are shown here. The first was a positive control in which uncontrolled transient expression occurred. So this vector shown at the bottom right corner of the screen is the positive control vector. And this allows researchers to have a benchmark for what would occur when there was no controlling switch in the plant. And this was kind of based off of previous research, which has proven that this vector works in continually expressing systemic acquired resistance once the edited gene is put into the plant. The negative control, not shown here, only had a luciferase gene on it. And I'll get to that in a second, but it did not affect the immune responses in the plant at all. So the plants in the negative control group have immune responses identical to those that have not been genetically modified. And the purpose of the luciferase gene was to make sure that the edited genes had been taken up into the plant. So if the luciferase gene was being expressed in the negative control plants, we would know that the edited genes had been taken up into the plants. The experimental group, as described previously, is the one that uses a heat shock in order to activate systemic acquired resistance. These genes were built using Golden Gate assembly, a modular assembly method on a software called Benchling. The modular assembly method takes these gene sequences and then combines them into a vector as shown here in a more simplified manner than other strategies in the field, which is why we selected this one. Here is a close-up look of the experimental vector. I think there's just a slight lag in the slides, which is why it's not being shown, but oh, here it is. And you can see that the ALD1 and FMO1 markers are at the bottom left of the circle, and the luciferase gene right here, if you can see the cursor, is marked right there. So the second step was to obtain the actual DNA, which was ordered from a company called Twist Biosciences, and then sequenced using Sanger sequencing in order to verify the integrity of the product and make sure it was actually what we ordered. Afterwards, the physical DNA was transformed into E. coli cells using a freeze-thaw method, which we selected due to its previously proven high efficiency methods. Afterwards, we performed primary inoculation on the plants in which the edited DNA was inserted into the plants. In the top right, you can see the edited DNA being put into a plant. And in the bottom left corner, you can see the actual plants after they've been transformed with the edited DNA. So once the edited DNA was put into all of the plants, we put all of the plants, positive controlled plants, negative controlled plants, and experimental group plants into a heat shock chamber. And we did this because even though the heat shock would only affect the expression of genes in the experimental group plants, we wanted all plants to have identical conditions throughout the experiment. We then performed the luciferase expression test to make sure that the edited genes were being expressed in the plant. So about 5 to 9 days after primary inoculation, we performed secondary inoculation on the plants in which we exposed them to bacteria and then harvested those leaves and plated them on solid media in order to count the bacterial colonies that grew on them. The more the colonies that grew on these plates, we could infer that the immune responses were not as strong. So we got 2 primary sets of results from these experiments. The first set of results, which was from primary inoculation and the luciferase gene expression test, shows that there is light being emitted from experimental plant leaves. And this shows that the luciferase gene was being expressed, and thus the edited genes being transformed into the plants was a success. The second set of results came from secondary inoculation, where we counted the mean number of bacterial colonies per experimental group and performed statistical analysis to see if there was a significant difference of means in the bacterial colonies that grew on plant leaves from each experimental group. And the statistical result was that there were no significant differences in the bacterial colonies that grew on these groups. So what this would typically mean is that we cannot control systemic acquired resistance through a heat shock activation. However, an error analysis actually suggests that an error occurred during the experiment, which might cause these results to have to be reevaluated. We can rule out a few possible sources of error, in which were the gene construct that was virtually made and the ordered gene product. And we can actually identify that the error was in the inoculation steps. Specifically, we left too much time between primary and secondary inoculation, such that the edited genes had been naturally cycled out of the plant's expression, and they were no longer being expressed by the plant when we inoculated them with the bacteria. And this just means that all the plants no longer had the edited DNA being expressed within them or able to be expressed within them. And so all the plants were, for all experimental purposes, identical and had no differences in their ability to express edited genes. So since these conclusions have not been able to be supported by the experimental process, we suggest future research in which we repeat this experiment, but rather than leaving 5 to 9 days between primary and secondary inoculation, decreasing that time to be maybe 4 to 6 days, so that the edited genes can be expressed when secondary inoculation occurs. Furthermore, we can also suggest future research in changing the immune pathway. So instead of systemic acquired resistance, some other pathway, changing the target in general. So instead of an immune pathway, maybe frost resistance or drought resistance, changing the external stimulus instead of a heat shock to be a cold shock, or a completely different experiment in which transient expression levels in plants would be quantified and recorded. So researchers could have a way to see how they change throughout a period of time. And this would help future projects in this field from having to meet the same difficulties as this one. Thank you all for your time, and I'll be glad to take any questions you might have.
Absolutely. Nice job, Advait. So it's time for your oral defense questions. And at this time, you've had an opportunity to prepare for these oral defense questions. And I'm going to ask you one from each category: the research process, your depth of understanding, and reflection on the inquiry process. So my first question for you is this: how did your initial exploration of the scholarly conversation lead to your final research question?
That's a great question. So throughout my research process, I always wanted to do something that would have a real life impact. And using genetic engineering to help increase crop production was the overarching goal that I always had from the beginning. However, there were multiple ways I could address this problem, such as using genetic engineering to confer frost resistance or drought resistance to plants. I looked through these avenues and kind of planned out to see if this would be a possible experiment that I could do. And looking through these possibilities, I was able to rule out a lot of them because of the extent to which previous research had already been done. And the research that would still be novel would actually take too long to fit into the given time period for this class and AP research. So looking at pests and pathogens, I noticed that they had a really large environmental impact as well as an economic impact. And I also noticed that there were some novel opportunities for me to do research because they hadn't been explored as in depth as the other opportunities. And so through this line of reasoning, I chose to do genetic engineering applications in combating pests and pathogens in agricultural scenarios.
Great. Tell me a little bit about the limitations of your method and how that influenced your new understanding.
Yeah, so there are actually two main limitations to the methods that I can identify from my project. The first of which was the increased duration of time between primary and secondary inoculation. Because of this, we cannot confirm the conclusions of the project, and this study would have to be repeated with a changed, more specifically, decreased duration of time between primary and secondary inoculation in order to confirm the results. However, a second limitation was the sample size of the project. So this research project used smaller sample sizes in which 3 experimental plants were used in the experimental group, the positive control, and the negative control. And the small sample size allows for uncontrollable factors to have a larger effect on the results of the project. So this must be taken into account when looking at the results and conclusions of this project. And the small sample size limits the extent to which we can apply the conclusions.
That makes sense. And then my final question for you is, what was the most important research skill you developed as a result of this process, and how might you apply that to future endeavors?
Yeah, I think the most important research skill I learned in this project was dynamism and adaptability. So throughout this process, I used a planning method called the dynamic expansion project planning model. And what this means is that as we go through the project, we come up with an outline of large goals. And these goals wouldn't be detailed. They'd be goals that we want to achieve in the project as we go. And then under each goal, we would have smaller, more specific goals, sort of steps in the project that we would complete. And this method allowed for flexibility because we would fill in those smaller steps as we continue the project. And I think this was definitely something I would use in another research project because it allowed for changes in scheduling, changes in availability of plants, and the availability of the resources to be taken into account when designing the experiment. And it also set us up to be more ready for the uncertainty that comes with the project because the flexibility allows us to adapt to any unknown changes that might occur. So I think being able to be flexible and change our practices based on possible uncertainties is one of the biggest skills I learned through this research process.
Outstanding. Well, that finishes the formal part of your presentation with oral defense. I'll stop recording here.